Hello and welcome to Bio40 Pre-Lecture 28. We've been talking about gene regulation in prokaryotes. Now let's see how it works in eukaryotes. As important as proper regulation is in single-celled organisms, it has an even bigger role in multi-celled eukaryotes. The various cell types in your body all have the same set of genes. A heart cell is only different from a brain cell because different subsets of genes are active at different times. In fact, there are hundreds of different cell types that are all the products of tightly controlled gene regulation. These are all the steps we talked about that lead to the expression of eukaryotic genes. In bacteria, we mostly focused on transcription and translation. And we did talk about post-translational modifications a few lectures earlier. Eukaryotes can regulate these steps too, but they also have post-transcriptional processing, export of the mRNA from the nucleus to the cytoplasm, and the localization of proteins to the proper subcellular compartment. Each one of these steps can be regulated to make sure a given protein is only present and active when it should be. Additionally, chromatin structure in eukaryotes provides another level of regulation. Chromatin is a term for the combination of chromosomal DNA and the structural proteins involved in packaging. Way back when we discussed the debate about whether DNA or proteins are the genetic material, I mentioned that our chromosomes are about 50-50 of each type. We established that DNA is the genetic material, and now let's finally take a look at the proteins. These proteins are called histones. They are small and positively charged, which helps them bind the heavily negative DNA, leading to a net neutral charge. There are 5 different histone proteins: H1, H2A, H2B, H3, and H4. Those last 4 make up the core of a key structure called a nucleosome. The nucleosome is an octamer, consisting of 2 copies each of histones H2A, H2B, H3, and H4. You can see the disk they form here, and if we could rotate the image 180 degrees, you would see another copy of each of the proteins forming a second disk. 160 base pairs of DNA is wrapped around the nucleosome in 2 loops, and there's a 40 base pair linker between adjacent nucleosomes. The last histone, H1, interacts with both the core proteins and the linker DNA. The chain of nucleosomes forms this beads on a string structure, with each nucleosome having a 100 angstrom diameter. And then there is supercoiling that occurs, which leads to an even more compact structure with a 300 angstrom diameter. So our DNA can be quite condensed. This condensed structure can leave the DNA inaccessible to proteins like RNA polymerase and other factors needed for gene expression. We use the term heterochromatin to refer to DNA that remains condensed throughout the cell cycle. This includes telomeres and centromeres that don't have genes to be expressed. And this is also how exon activation occurs in women. Other parts of chromosomes can go in between compact and non-compact forms. DNA that isn't tightly compacted with nucleosomes is more sensitive to being chewed up by enzymes like DNase. And in vitro studies, DNA sensitivity is correlated with transcriptional activity. So being in a less compact form makes a region of DNA more likely to be transcribed. Modifications of histones H3 and H4, including methylation, acetylation, phosphorylation, and ubiquitination, influence which form the chromatin takes. Histone acetylation in particular is associated with less compact states and more transcription. So what are the regions of DNA that need to be accessible for transcription to occur? Just like bacteria, eukaryotes have promoters, sequences where RNA polymerase binds and initiates transcription. Eukaryotic promoters can be highly variable, but the most common is the TA-TA box, a region rich in AT base pairs. How well the polymerase binds the gene's promoter gives a basal or baseline level of transcription. But there are other factors that can increase or decrease how much transcription occurs. Enhancers are analogous to operators in bacteria. They are DNA sequences where proteins bind and affect the level of transcription. But unlike operators, enhancers do not need to be close to the promoter. and can in fact be thousands of bases away. They can be upstream or downstream of the transcriptional start site, but they must always be on the same piece of DNA as the genes they regulate. They are cis-acting elements. Transcription factors are the proteins that bind promoters or enhancers and either help or hinder polymerase binding, though this influence isn't necessarily direct. Sometimes the transcription factors recruit other proteins, and these are what interact with the polymerase. As with prokaryotes, there are activators and repressors, though there's a third kind of transcription factor called basal factors. Basal factors bind the promoter and are needed to achieve the baseline level of transcription. The TA-TA box binding protein, or TBP, binds the TA-TA box. Then, TAFs, or TBP associating factors, associate with TBP. RNA polymerase II binds the TAFs and transcribes the gene. Activators are proteins that bind enhancers and interact with basal factors to help recruit the polymerase. This interaction can be direct or indirect via a mediator protein. Note the way the DNA curves here. This is how an enhancer can be hundreds or even thousands of bases from a promoter and still influence transcription. Though this isn't the only way that activators function. Some recruit other proteins, called coactivators, that help displace nucleosomes. This can make a promoter accessible to the basal factors and polymerase. Conversely, repressors can block basal factors and RNA polymerase from binding the promoter, or they can encourage nucleosomes to form closed structures, leaving the promoter inaccessible. This is often done with the help of another protein called a corepressor. Note that this term means something different in prokaryotes and eukaryotes. In bacteria, we said a corepressor is a small molecule that binds a repressor and helps that repressor bind the DNA. In eukaryotes, the term refers to proteins that are recruited by the repressor after it's already bound the enhancer. Additionally, there are indirect repressors, which don't interact with basal factors or histones at all. Instead, they interfere with activator function, and they can do so through various means. In competition, the repressor binding its site blocks the activator from binding the enhancer. Other methods work through direct interaction with the activator. In quenching, the repressor binds the activation domain, which stops it from being able to interact with basal factors or histones. Cytoplasmic sequestration involves binding the activator in the cytoplasm and preventing it from reaching the nucleus. Finally, some transcription factors function as homodimers, and the repressor can bind one monomer to prevent dimerization with the other. A given transcription factor can act on multiple genes, and it doesn't even need to play the same role in each case. This transcription factor dorsal can serve as an activator and recruit a coactivator to increase the expression of one gene, but it could dimerize with another transcription factor to recruit a corepressor and lower transcription of another gene. Though this isn't always the case, eukaryotic transcription is so tightly controlled that often a transcription factor is expressed solely to regulate transcription of a single gene. There are about 2,000 genes that encode transcription factors in humans, accounting for nearly 10% of our genes. There's another fascinating way transcription can be regulated called epigenetics. Eukaryotes sometimes add a methyl group to a cytosine that is followed by a guanine. This simple sequence is called a CpG motif. There are some regions of DNA with a high concentration of these motifs called CpG islands. Another way activators can function is to bind CpG islands to prevent their methylation by methyltransferases. Histones don't bind unmethylated islands, leaving the chromatin structure open and the promoter accessible for transcription. But when there is no activator present, the CpG islands are methylated, and methyl CpG binding proteins bind and cause the chromatin structure to close, so the promoter is inaccessible and there is no transcription. The reason researchers are so interested in epigenetics is that the methylation patterns are inherited. After the DNA is replicated, DNA methyltransferase makes sure the new strand has the same methylation pattern as the old one. Gene expression affects phenotypes. So this is a way that traits can be inherited that is independent of the base sequence of the DNA. This comes into play with a phenomenon called imprinting, where the expression of an allele can differ depending on whether it's inherited from the mother or the father. There are about 100 genes in humans with imprinting control regions. Methylation of these regions silences expression, as we saw before. If an allele is paternally imprinted, it is methylated and silenced when inherited from the father. The same allele from the mother will not be silenced. Likewise, maternally imprinted alleles are methylated and silenced when inherited from the mother, but not from the father. The imprint pattern is consistent throughout the offspring's life in its somatic cells, but is reset in the germ cells. In meiosis, the old methylation pattern is erased, and the new pattern is set. So a woman will inherit a set of chromosomes with maternal imprints from her mother, and a set with paternal imprints from her father. But in meiosis, everything is reset, and then all chromosomes get the maternal pattern. And in a similar way, when sperm is produced, the imprints are erased, and then given the paternal pattern. Okay, enough transcriptional regulation. Let's look at a transcript processing example. We talked about alternative splicing before, but here's a particularly interesting case. As you might expect, male and female flies have different mating behaviors, but those behaviors are controlled by the same gene, fruitless or fru. The difference is that females express proteins called transformer and transformer 2, which interact with an exon on the fru transcript. This causes that exon to be skipped over during splicing, leading to the female variant of the fru protein being expressed. But in males, there is no transformer protein expressed, so all exons are included in the final transcript, and the male variant of fruitless is made. Finally, let's talk about a way eukaryotes stop mRNA molecules from being translated. RNA interference, or RNAi, involves short pieces of RNA that base pair with mRNA. There are several types of these short RNAs, including microRNAs and small interfering RNAs, which we'll discuss. There are also Pee-wee interacting RNAs, which are less well understood but seem to play a role in reducing transposon movement. MicroRNAs can be transcribed from non-coding regions, or can be found within the introns of genes. They have inverted repeats that form stem loop structures within the transcript. Such transcripts are called primary microRNAs. The enzyme DRASHA excises the stem loop from the transcript. The excised stem loop is now called pre-microRNA, and it is exported to the cytoplasm. There, the enzyme dicer trims the ends of the pre-microRNA, leaving a mature duplex microRNA. The RNA-induced silencing complex, or RISC, takes one strand of the duplex to use as a guide. When this guide microRNA in the RISC pairs with an mRNA, translation is prevented. This can happen in two ways. If the microRNA perfectly base pairs with the mRNA is cleaved and then degraded. But if pairing is imperfect, so most but not all bases pair properly, the mRNA is not cleaved and degraded, but RISC binding still blocks translation from occurring. Small interfering RNAs, or siRNAs, are very similar to microRNAs, but they originate from double-stranded RNA rather than hairpins within transcripts. Double-stranded RNA can be made from transcribing both strands of the DNA, or be introduced by viruses. Dicer processes the long double-stranded RNA into smaller fragments, and then one strand is incorporated into the RISC, which operates as we just saw. Cells can use this to downregulate expression of their genes, but this is also a line of defense against viruses by limiting their gene expression. Additionally, researchers can intentionally add double-stranded RNA that matches a sequence of a certain gene to knock down the expression of that gene. Targeted knockdown of specific genes is important for a lot of research, and the discovery of this system earned Craig Mello and Andrew Fire a Nobel Prize in 2006. Well, that's the end of gene regulation, and actually the molecular genetics unit overall. Next time, Eco-Evo students rejoice, it's population genetics.